Faranak Farzan, who is an associate professor at SFU and the chair in technology innovations for youth addiction recovery and mental health. You'll hopefully learn today that her research is very interdisciplinary, spans topics like engineering, neuroscience, computer science, and medicine. And her work really focuses on neuroengineering solutions to diagnosis and treatment of mental health. So turning it over to you, Dr. Farzan, and looking forward to your talk.
Thank you for that introduction. So we are Center for Engineering-Led Brain Research. So for my colleagues in the room and also students, if they want to pop by and see the kind of work we do, we're positioned and we're placed in Surrey, next to Surrey Memorial Hospital. So we have a decent sized space where we are equipped with all kinds of neuromodulation and electrophysiology equipment, where you can come by and see the kind of work we do. It's very close to the imaging center of SFU as well, and that proximity with imaging and MEG can also be very advantageous from a collaborative perspective if we decide to run studies together. And I hope by the time we go through my idea talk, we have some ideas for collaborating on a grant or on a project. I'd like to give a shout out to my team at SFU and all the students who have worked with our team over the years, many of whom are in this room, a good proportion of the room, which makes me happy. So come and say hello to us in SFU and check us out if you happen to be in the area. In addition to the local work, we do a lot of international and national collaboration, including with Jane Foster from Combined. So Combined is also a big pan-Canadian network through which we run quite a few clinical trials in patients with depression, going through all kinds of clinical trials. And we're in the business of biomarker discovery for depression. So we collect, for instance, electrophysiological data from all these patients. One of our students in the room is in charge of managing all that EEG, Benjamin here, standardizing it, and then coming up with biomarkers of response to various kinds of treatments. And we do work with Jane also, hopefully, even more so now with connecting some of these brain markers to hopefully got a microbiome as well, and all kinds of other collaborations as well, which I think is important to highlight. I won't repeat what Paul set the tone for, but just for us to be contextualized and in case people on the Zoom are coming in and going out. So there is a heterogeneous definition for resilience. So for the purpose of this talk, so resilience refers to the mechanisms that empower an individual or an organism in general to withstand the impact of challenging circumstances or trauma without falling ill or suffering from mental illness. And Paul did a very good job of also telling us and all the previous speakers about all kinds of brain structures that might be involved in the process of resiliency. So I won't repeat that neither. Just in short, limbic structures, including amygdala, anterior cingulate cortex, prefrontal cortices, they're all thought to be involved in the process of resiliency. Now, what would be very interesting from my perspective in the spectrum, in the sphere of biomarker discovery, is to be able to look at somebody's brain here and say whether that person is resilient or not. So we're getting maybe sort of into this biotype perspective, but with a different tool to play with. So there is a technology called transcranial magnetic stimulation combined concurrently with electrophysiology. You can shorten it for TMS-EEG, which we've been developing in my lab and in collaboration for the past 10, 12 years. So it looks like that. So so Paul talked about the coil, but what we can actually do with that coil is pair it with electrophysiology concurrently. So now you can give a pulse to the brain, kind of like a perturbation, or you can think of it even if we compare it with the animal literature in which a shock is given to an animal and you wait for the response to that stressful situation. You can think of it that you're stimulating the brain with a pulse and you're seeing, how it's going back to its normal state again. And maybe we can use that as a proxy to look at how resilient someone's brain is. and whether that can help us predict future occurrence of mental illness or in general coping with stressful situation to begin with. What is cool about TMS-EEG is over the past, again, 10, 20 years, we have come up with actually the underlying neurobiology of it too. So you can think of TMS-EEG in two ways. You can think of it as a tool where you can actually probe the brain and look at the reactivity of brain to it. So you're providing a stress relief because this is a big, giant stimulation that is happening to the brain. And then you can look at its reactivity to this stimulation. So you can look at the response, brain response to the stimulation. Or you can actually come up with protocols using single pulse versus also paired pulse protocol, where you can look into the circuitries, and pinpoint the exact neurotransmitter system that you're studying. So some of the work we've done along with others over the years have allowed us to actually link some of these responses to the brain stimulation to very specific circuitries that I'm mentioning out there. So when you give a small single pulse, a supraterrestrial pulse, meaning it creates an action potential that starts going around in the brain, you see this response that looks what I have on the curve on the slide with these prominent peaks with very specific latencies that you see. So a latency of a peak may happen around 15 milliseconds after the pulse, one at 30, one at 60, one at 45, one at 100, and one at 180. And each of these peaks and troughs through pharmacological manipulation have been shown to be linked to specific circuitry. Either it's slow-acting GABAergic system, such as N100, or it could be fast-acting GABAergic system with GABA-A receptor-mediated activation with N45, or it could be more excitatory waves like the P15, P15, N15, P30, and maybe also the P180. So these are linked to the excitatory system. So quite a few pharmacological studies over the years have shown that if you blockade those receptors, those peaks would be modulated. So here we have a tool which we can again use in two ways. So I repeat, so you can look at the reactivity and you can look at brain circuitry. So this has allowed us to actually take a lot of animal studies and bring it into awake humans and be able to look at those studies. And that makes TMS-EEG a very interesting tool for cross-species studies as well. So you can actually, there are TMS coils, by the way, for animals too, and you can do some of these studies in animal models too. It's not as straightforward as in humans actually, but you can do that too. So you can actually work across species. So now why am I telling you about this and what does this have to do with resilience yet? So in my lab, we work a lot with biomarkers and biomarkers of depression, such as in youth. So some years ago, 5, 6 years ago, I wrote a grant. I was very interested in looking at what could be a neuromarker of youth depression, because particularly in youth, a good proportion of them, 50% of them, do not respond to treatments. Or they do not want to receive first-line treatments because of some of the side effects. Just to give you an idea, SSRIs have FDA Black label on them, meaning in those 24 years of age or younger, they could get, it could actually exacerbate suicide ideation, which seems counterintuitive. So there are many good reasons for us to be looking into the brain stimulation literature specifically for youth with depression. But not a lot of work had been done at the time in this particular group. So we decided, why don't we actually use TMS-EEG to look at some of these neurocircuitries between healthy youth and youth with depression, and we're talking about an age range of 15 to 24. So really right around the time that the brain is developing and when the onsets of lots of psychiatric disorders are. We had a specific hypothesis, so there was a lot of literature already on DLPFC. I won't bore you with details because Paul went through it quite extensively. So we wanted to stimulate prefrontal cortex to see, and we had this hypothesis that the prefrontal cortex reactivity would differentiate youth with depression from healthy youth. And we throw in the mix some control brain areas just to see how specific that particular finding could be. So we stimulated also the default mode network, and we also stimulated sensory motor areas. So how did the experiment look like? So again, you hold a TMS coil over area of interest, you stimulate, and you measure these dynamics. So with a very high resolution. So again, those waveforms that I showed you, you can recover the signal as early as 15 milliseconds after. And this topograph here is showing the evolution of and the traveling of that signal through the brain structures. So you can then do this for every brain area in healthy participants, and then make that comparison with youth with depression. So we saw something really quite strong as a finding. So we saw that actually very specifically in the left and right DLPFC, we had this prominent response in the late waveforms, in the N100 time point and P200 waveforms. That was quite differentiating between healthies and youth with depression. This is the work that Paul did under my as a PhD students. Under my supervision, and we looked at source localization, and these sources tend to be, well, again, EEG is not the best method for understanding where things are happening. It's great for time, but the source localization do suggest that these effects were rising from anterior cingulate cortex and superior frontal gyrus. Some of the areas actually linked to resiliency as well. Which I will get into once I get into the actual idea that I have. So you may say, okay, there's some overreactivity in these regions, but are they clinically meaningful? So in fact, this over-exaggerated reactivity that we saw around 200 milliseconds after the pulse, which presumably is related to the excitation inhibition balance, seem to correlate quite strongly with the anhedonia measure. Which was interesting. So it seemed to actually have a relevance to clinical symptom. Now, and Randy asks me to give a talk on resilience, and I go searching. Has anyone done TMS-EEG and looked at resiliency versus people who are vulnerable and people who are resilient? And in fact, a very recent paper during COVID-19 had actually looked into this. And they had a very interesting finding that ties in really well with what we saw even in depression. So they had a hypothesis. So first of all, they took TMS as a toy model of studying resiliency, which is, again, you have a very simple model here and try to understand how the system responds to this supposedly stressful event to the brain. So they took, this is a group from Barcelona that had a cohort study at a time running, and it coincided with the wave of COVID-19. So they had these folks who were being followed up with their depression scales over time, and they happened to have their TMS-EEG right before the pandemic hit, just a few months before. So they had an idea that potentially the DLPFC, the frontal parietal network, would be in some way linked with resiliency, and they had picked a node in default mode network, IPL, as their control site. So what they found is in fact interesting. So they found, very similar to what we showed with youth with depression, this over-exaggerated response around late response around 200 milliseconds in those who later on during the confinement of COVID-19 came down with symptoms of depression and anxiety. Not quite as much to cut the clinical boundaries, so they did not become clinically depressed or anxious, but it was enough to be able to differentiate the two groups from the symptom perspective. So I find this, and I find this quite interesting, that perhaps maybe part of the component that we're looking at, also comparing youth depression with healthy, is a component of resiliency, because these two waveforms are exactly coinciding with each other. Now, the question then becomes, so potentially maybe those who are more resilient to more resilient, they are able to recover maybe faster or in a more stable way from a single pulse of TMS. So that could be one idea, and we can differentiate them and biotype them that way. But the more interesting idea, which Paul was also getting into, is that can we modulate it? So now we've got this overactivated prefrontal response. Is there a way to modulate it and maybe make people more resilient? Is resilience something that is stable, or it can be actually modulated across our lifespan with interventions such as brain stimulation? So this is where the idea talk comes from, or gets seeds in. So we've done 2 clinical trials in youth depression, following the findings that we had with this over-exaggerated left and right prefrontal cortex reactivity to TMS. So one was a 2-week RTMS treatment in youth with depression, and one was a 4-week combined with cognitive training RTMS clinical trials. In both trials, we saw quite an interesting modulation of TMS-EEG response after the treatment that was linked with reduction in symptoms. There were several questions from Paul earlier about the efficacy of brain stimulation potentially. And the trial that I'm showing, the modulation of the signal on the right, was actually a very successful trial in which 76% of participants actually recovered. So this is in youth combined with cognitive training, open label, very, very, preliminary way of looking at RTMS for youth in particular. But the take-home message is there were some modulations of these TMS-EEG responses that correlated with behavioral change following RTMS treatment. What is also very interesting, because we know that limbic system and potentially default mode network is also very much involved in resiliency. So I want to highlight that on the trial on the right, on the left side, what I'm showing you is actually some downstream changes in the parietal area. So even if we stimulated the prefrontal area, we saw modulations of response to TMS after 4 weeks in the parietal, in non-stimulated areas, which again linked with changes in symptoms. So which goes to highlight that, well, this is a highly connected network that we're stimulating, but also perhaps there are changes in maybe in the areas that cause depression or more cause underlie resiliency. All right, so I think I went over time a little bit, 1 minute.
It's okay, we started a bit late.
So we can use the rest of the time to discuss the idea of TMS being a model for resiliency and using the biomarkers of TMS-EEG to develop treatments for resilience.
Thank you, Dr. Farzan. That was a fascinating talk. I'm going to open the floor for questions. Oh, go ahead, Randy.
Great talk. I got a question having to do with, it's kind of getting back to the mechanism question I asked Paul as well. And when you showed the difference, for example, in the COVID paper, and what you showed in the training as well, suggested there could be 2 things, at least, that are happening with the response to TMS and the changes that have to do with resilience or not resilience or risk. One could be that the system becomes less deterministic. Mm-hmm. So, for example, just to explain what I mean, if you stimulate an area and all the other areas it's connected to respond the same, essentially, that's more deterministic. I'm saying this for the audience, not necessarily. This is Randy, by the way. However, if there's more stochasticity, then when you stimulate, the areas may not respond all the same way. They may actually just respond on their own. There may not be quite as strong a response across the entire brain, which means it allows it to essentially explore more opportunities to get back to the repertoire idea. The other option, of course, is that the areas become less responsive. So it's not so much that there's a change in the deterministic versus stochastic, they just become less responsive. Mm-hmm. Do you have an idea of which of those 2 it might?
Yeah, very interesting.
If that's actually the only 2.
I'm looking at one of my other postdocs here in the room, Chloe, who is actually looking at that as we speak. Right. So when you look at, so with TMS, you can perturb the system. You can also do all kinds of, you can look at the brain response in all kinds of different ways. So one way is to look at how complex the response to this stimulation is. There are work from sleep studies that actually has looked into that. So if you stimulate the brain during wakefulness, you see a much more complex response with those peaks and troughs that I showed you. But if you stimulate the brain during sleep, for instance, when some of these areas are actually disconnected from each other, you see a more simple response and usually higher in amplitude. So clearly, what you, as you mentioned, you could be looking at the complexity of the response as a proxy of the system state in some ways. And Chloe, in fact, here is looking at that, is looking at all kinds of TMS, single pulse TMS-EEGs in the world that's been collected out there and looking at that. And also the idea is also whether you can use TMS to modify, or in some ways, the complexity or the amount of information flow in the brain as well. Does that answer your question?
Well, it's a question of which option takes time.
I think there is a different configuration. So depending on the configuration of the brain, you may have less complex or more complex response. So I don't think it's less responsivity. I think it's just a different network configuration that potentially is less perhaps tailored for processing information. Would that make sense? That would be my guess. So it's responsive. It's not that it's not, but it's just not as complex. So maybe on the far side of the spectrum, you would have pure synchrony. Right. So it's somewhere, so that you are somewhere between the spectrum of, and I would argue that we have done some work in complexity also in the context of seizure treatment. So, and we've shown that actually it's time scale dependent. So maybe at a local, from a local region perspective, you may have over exaggeration of complexity, but in the longer range format of the interaction, that might be, less complexity than there is. So I would argue that this is more complex than that. So it could be a matter of long range or short range and local or global. And
okay. Hi.
Hi.
My name is Shakiba from SFU.
Nice meeting you.
So I have a question about, I have two questions. The first one is about so you showed a topograph on the first one that you showed, it was moving. Yeah.
I showed
the one
moving
before
things.
this.
The other one? Okay.
Yeah, that one. So is this one TMS signal that's this entire thing?
Yes.
Okay, so this is not different areas. This is just
This is one single pulse. So there is a timer there. I'm not sure how visible it is. The timer there is showing how far from the pulse you're seeing the evolution of the signal.
Right.
So if we can wait for it to reset. So this is at 140 millisecond, and it should very soon go back to 0. So if you let me wait for the 0, because at the 0, you actually start seeing a local response, which is very confined to the area of stimulation. And now, wait for it, I think goes up to 300. There you go, here. So it's very local, it's very fast at the beginning, and then it becomes a slow, right? And then it sort of stabilizes. Yeah. So you can look at connectivity, right? Because clearly things are not staying local. It's moving really fast. And there was some local circuitry involvement too, as I point out here.
Awesome. Yeah, that's really interesting because I love TMS and I love EEG, and this is just so cool to see. And then my second, sorry.
I'm so happy to hear.
My second question was, so you worked with youth, and I'm wondering what age range exactly that is, and if maybe from 12 to 14-year-olds versus 16 to 18-year-olds, maybe this would be different because of the way that the brain is developing, and or like if the differences just like cancel out because of the way that like the control areas are and everything, or or if there's differences.
Good question. All questions are good, by the way. So we looked at 15 to 24 in this particular. The reason for that was clinical. That's an age group in which youth are transitioning between psychiatry, going from pediatric psychiatry to general psychiatry. And so there was a clinical reason for choosing this age range. We in my group haven't looked at younger than 15, but there are other groups in the world, especially in Finland, that they've actually looked at in the context of ADHD. So they've looked at this TMS-EEG responses, and there are changes in the inhibitory component of them in youth, in children, right? And in general, children tend to have a bigger response around N100. It could, again, back to the question of Randy, it could be linked to the connectivity of the brain. Perhaps they're not as much crosstalk between all brain regions when you're younger, and then the older you get, one of wisdom of age is really maybe more connectivity and more complexity in information processing across. So the TMS-EEG neuromarkers here could actually be looked at over time to sort of see that neurodevelopment as well, and potentially neurodegeneration with Alzheimer's as well.
All right, we have a question from Robert on Zoom. When you say that the response to TMS becomes less complex or more complex, does this map onto the idea that Randy posed with this question of responses being deterministic? Would a less complex response be more deterministic and vice versa?
No, yeah, that's a great comment. Yeah, I think so. Yeah, it's an easy one.
I just want to follow up on a question that was asked just a little bit ago about different age groups. I'm just curious if the pattern of cognitive differences and symptoms and brain patterns look very different in folks who are diagnosed with child depression versus adult. Like, just if your depression comes online when you're very young, and now apples to apples comparing, like teenagers or adults, is that a different type of depression? So what we're learning from this work in this group, can we extrapolate that to other groups, or do we really need to think about child onset versus adult onset depression as very different things?
So I have to say that TMS-EEG has been explored in adult depression as well. And so a lot of these findings are mapping quite nicely also to the adult depression. Now, going to younger groups, younger than 15, so looking at children, that might be a different story. And also, again, because the TMS response in a child brain could be also is different in terms of amplitude, not necessarily the shape perhaps, but in terms of the magnitude of response or perhaps the complexity of response. So there hasn't been a systematic study looking at all groups together just to be able to give a comprehensive picture of what's happening across age. But there seems to be quite a bit of continuity, at least between the work we do and the adult depression. In fact, for instance, this overreactivity of the prefrontal area that I showed has been shown in adults, which mean age of 45, which is like our mean age is 20. So it's quite several decades ahead. So
Thank you.
Yeah.
So I was really interested in the finding that you mentioned about sleep versus wakefulness. And I think you said during sleep, there's a less complexity of perturbation. And I'm just curious, what's the relationship here with EEG's coverage, right? Because there's a pretty good coverage over surface regions, but it gets weaker and weaker, and up to the point of becoming more or less blind to deep subcortical structures like the hippocampus, for example. So, and we know from fMRI that during sleep, the frontoparietal control network shuts down. It's actually actively deactivated, right? It's suppressed. but places like the mediotemporal lobe are actually more active during sleep than during wakefulness. So I'm just wondering if you think if we could see the electrical, or maybe someone has done studies with implanted, like patients with implanted electrodes, to see whether there might be a flip between response to perturbation, so that limbic structures, subcortical regions might show the opposite pattern to what cortical structures might show to perturbation. And because that will have a big relevance to depression, right?
Yeah.
To the extent that we think limbic regions might be some of the leading causative things like anhedonia and all these very fundamental effects, probably not cortically driven.
Yeah, absolutely. I'm looking across the room to see whether I see my student who works actually with that. Raj is in here, right? No. Randy was this examiner on Raj's work. So we do have a collaboration with Iowa University with a group of neurosurgeons who actually do have implanted electrodes in patients with epilepsy. And we're supporting them and working together on this framework of TMS, EEG, and IEEG. So looking at the whole spectrum of things. Work in progress, but absolutely, these are some of the questions that we're also interested in. I'm not sure how feasible it would be to actually look at that in sleep, given the complexity of the situation, and then the burden to patients, particularly. Getting TMS during sleep is pretty awful. I'm personally very interested in sleep studies, but there are also some questions about, hey, look, when you stimulate somebody during sleep, are they still asleep? Are they not? State of sleep that, there are all kinds of control conditions. And I think the sleep studies with TMS are pretty neat actually. Yeah, I'm a Simuni group and and the Wisconsin group, Tononi, yeah. So it's pretty neat framework, but it's also very hard to do. It was a science paper back in 2005 where they actually showed this and the idea of. So we're looking hopefully at some of that work. Again, these are patients with epilepsy, so so that has to be taken into account. But absolutely, so there's some challenges at the methodological level in concurrent combination of all these methodologies from signal recovery perspective. We spent a good maybe a decade or more just trying to figure out how to recover the EEG after TMS. So a lot of studies that have happened before, now they're being redone also because now we're able to recover more part of the signal and then look at earlier signals as well. But those are exactly the right questions to answer and go about next. Yeah.
We have another question from Zoom from Anvita. How long does the effect of a TMS pulse last? And if I understood correctly, you said it kind of wears out and gets more global. And also in the last talk, how long would the pulse or such impulses of excitation or inhibition be?
Yeah, very nice. So here, I think I stopped the timer on this particular topo plot around 300 milliseconds. And the reason for that is it takes about like the response that you see that is so distinct from the spontaneous activity usually lasts about 300 milliseconds. We've done the studies actually back in the days in schizophrenia, actually, that we showed in some of the patients. Actually, there could be a recurrence of a response way down the line, like after 300 milliseconds, that could potentially again go back to this idea of resiliency and maybe changes in brain connectivity as well. So it takes about 300 milliseconds. Okay, I guess for the future, I will extend that to show you that actually stabilizes because it's stopping at 300 milliseconds now. And the second question was excitation. What about excitation and inhibition?
How long would the impulse excitation or inhibition?
Yeah, so generally the TMS pulse, right after TMS pulse, you can think of it excitation, inhibition, sort of excitation cascade. So you've got a very fast excitatory response. If I was to stimulate the motor system, for instance, you would see a twitch in your thumb. And then after that, so some fast inhibitory pathways just fall into the picture where they're mediated by GABA A receptors. And following that, you get the long inhibitory pathways that kick in, which is what we see around in 100 millisecond. And then there is another area of disinhibition, which is linked to this later response around P200. So some people think this P200 is related to excitation inhibition balance, which, as I showed, is what we show in youth depression to be overreactive, in those less resilient to be overreactive. And I remember from Jane's talk this morning, there was this idea of microbiome messing with the GABA system. So that's maybe another collaborative idea here where we can actually, I'm not sure if anyone ever has looked at TMS-EEG and microbiome, but I think that would be super cool if we can actually do that as well.
I have another question. This is Daniela. This relates to a question that Randy asked Kalina, so trying to make connections across talks. Is there any work in the field with respect to the brain state one is in before the TMS being predictive of how the TMS, what the response is? And I'm sorry if this was covered in any of the talks already, but just curious.
I'm looking at my other PhD student here, Amin, who's actually looking at that. His PhD dissertation is about statement dependency of TMS. So people have started looking into that. So we are toying around with some ideas of these microstates being actually predictive of the TMS response. And I'm not going to spoil it because he has a paper just about being submitted. So we do show state dependency. It has been shown before with other measures of signal features such as, you know, the frequency content or the phase at which you stimulate, the phase of the signal at which you stimulate the brain. So more and more in the field of brain stimulation, we're moving toward this understanding of state dependency for several reasons. Some of these waveforms that I show you are pretty reliable when you, for instance, do a test reliability. So that N100, P200 response, you more or less see it from one session to another. The earlier responses, which more excitatory, are thought to be more state dependent. So depending on the state of the brain that you stimulate, you may see more or less, and you can see that visually in the motor system. So if you're stimulating a motor system to get a twitch in your thumb, the amplitude of that twitch can change. So the question is, does that depend on the state of the brain? Exactly, right? So that's some of the work that we're doing. And the second reason is actually we're hoping by looking at this state dependency, you're able to configure, oh my God, there is a crow in the room. But she flew away. I think it took a cheesecake or something.
For those at home, a crow has joined.
Right. So you can actually tailor TMS in general better as a treatment methodology, because maybe this variability that we see in terms of response to TMS could be linked to the lack of controlling for state of the brain, right? So some cool work coming from our team, hopefully in the next 6 months or something, I will be happy to forward you the paper, or you can talk to Amin more to learn more about it.
Thank you. That's super interesting. I was thinking about that because you'd imagine that the groups depressed, non-depressed their baseline would be a very different brain state. So how they transition into the next one as a result of TMS would be very different, potentially the speed,
yeah. So that was what we were thinking. We're thinking, okay, so if let's say there are states, there are repertoires, or and assuming that maybe one of them is over exaggerated in depression, or people with lack of resiliency, right? When you do brain stimulation, you perhaps want to be mindful of that. Either stimulate that brain state particularly to try to dampen it, or avoid it to try to not potentiate it further, right? So there are all kinds of interesting ideas of frameworks we can develop here around the state dependency. So the field is working to all this closed loop brain stimulation, which would probably what I foresee for brain stimulation field in general, using EEG and imaging.
Question?
So in addition to states, or I guess we were talking more like brain states, but I was thinking more behavioral states. Is there like longer term behavioral factors that influence kind of your response? Because I guess this would be similar to maybe like medication when it's supposed to upregulate plasticity, but then depending on the environment, if you have like a positive environment, it has positive effects. But if you are in a negative environment, it can make things worse. So do you see similar things like that with TMS, or have they been looked at?
Yeah. Very, very good. So very good discussion. So two things I can mention here to answer your question. One of them more directly, and one of them maybe as a way to highlight another work, which another student here in the room has done, which is quite cool as well. So in the trial that I'm not showing the clinical trial here, but take my words for it. This right panel here is the change following this protocol, which was repetitive TMS, immediately followed by cognitive training. And we think that's one of the reasons where the response rate, 76% is a quite high response rate, right? Even if some of it is placebo, it still is quite high. And the reason we did that, we wanted to leverage brain state. So the idea is if you change, because with RTMS, unlike single pulse TMS, which only changes the brain response for 300 milliseconds, with RTMS, you're changing the brain state for a good hour, maybe hour and a half, again, person dependent. And so if within that period of time, you sort of leverage that state that you created, you may be able to get double your effect, right? So this is what we thought we will do. We will stimulate the prefrontal area and then have the participant during that time of heightened change to engage in a cognitive task, right? So that's one aspect, right?
Have you done any acute studies? So, you do a single session of RTMS and then have them engage in some sort of task or behavioral something to like identify maybe what's the mechanism that is leading to those long-term clinical changes?
Yeah. Have we done that? No, I don't think I've done it in my group. But RTMS plus cognitive training, what has been done actually, so I was mentioning to Kalina, so people have tried manipulating or modulating hippocampus with cortical stimulation through connected nodes. There's this study back in the days, I forgot the year exactly, but a few years ago, I appeared in Science also. So they stimulate the parietal node to get into hippocampus, and they did 3 sessions, pretty acute, not a treatment, and they show immediate effect on some sort of working memory performance. So those kind of work are out there where you can actually see changes. Yeah.
I think we're out of time. Thank you,
Alrighty.
Faranak. That was
Thank
a really
you.
interesting talk and discussion.